weekend. As you are painfully aware, there's an exam coming up over the weekend, but for most of you it will be concluding on Monday. We'll have 4 groups staggered every 30 minutes, proctoring starting at 6 over in the micro lab. I'd like for you to get a camera and help us out with this so-called pilot project. But anyway, you can sign up to take the exam with a live proctor. Don't need to schedule with ProctorU, and that will be available again. Also, this Saturday, I will give a review session on campus in the main seminar room in the microbiology building, starting at 1 o'clock and ending up around 3. And I don't read the exam or anything, but I'll go over any questions you have and any questions I can think of as we go along. I'll sort of make sure that all the high points are covered and that you'll be ready for the exam. I will have that available to participate by Zoom if you like. So if you're sitting at home someplace, at the beach, all those places you go on the weekends, you can still do it live. And we have a graduate TA who's a distant student who sort of manages all the questions and stuff. So you'll be able to ask questions just like last time. What else? We may get through a little early for this module. That's not to say that I want to cram more stuff in here, but I may have a chance on Thursday to do sort of a mini review and answer questions in class also. I think next year probably I'll segregate out the agrobacterium stuff and include it in Module 2, so we won't have so much stuff jammed up in Module 1 next year. Okay, so we've already begun a discussion on transposable elements, right? We were talking about the DNA-based transposons in eukaryotes, and I had some maize up here, and Barbara McClintock, and all that good stuff. And today we're going to talk about retro elements, retroviruses and retroelements. Retroelements is a kind of a general term which includes everything other than the viruses. The commonality in all of this is the prefix retro. Retro means backwards or going back. So this refers to the so-called violating the Watson-Crick sequence of events and information transfer from DNA into RNA. This is where you have RNA used as a template to make a DNA copy. And these retroviruses all use that in one form or another, hence the retro tag. And you see here the sections assigned are 15.1 is essentially the whole chapter. 15 is on transposable elements and retroviruses. We'll go all the way through the end. Some of the last topics we won't cover in depth, but you'll be responsible for what's on the slides here. So let's get started. Retrotransposons, and here you see a retrovirus, but retroviruses, of course, are not the only thing we have. Here you have up here this is an element in yeast called TY, and it doesn't make an infectious particle. And here you see an actual retrovirus. So this is a retrotransposon, actually, and we'll see the distinctions in just a moment. Retrotransposons versus retroviruses. The retroviruses, as I tried to point out here, I didn't read the slide, is forming an infectious particle that escapes the cell that's infected and infects other cells. That's the distinction. A retrotransposon has everything, it's the same, except the particle that it makes does not escape. It just is packaged, and then it's unpackaged and infects the same cell. Retrovirus-like cycle involves transposon-like events. And so you've got RNA, DNA, then RNA again, the same kind of thing you're going to see with the retrovirus. Reverse transcriptase, integrase, transcription. These two in sort of a green, blue-green, are activities that are encoded by proteins made by the virus. And you'll notice the last step is host RNA polymerase actually makes the RNA. This is a favorite question of homework and sometimes appears on exams. You got to tell me which enzyme does which step. So this is not hard, right? Integrase takes care of integration. What is that analogous to with a DNA transposon? Transposase, right? Reverse transcriptase is turning the RNA into DNA, and you have a linear DNA form, and then that linear DNA form is then inserted into the genome. You'll see that this linear DNA form is the only complete form of the virus. The other stages have bases missing. We'll see how all that works in a moment. The integrase step, the integrase cuts two bases off of each end as it inserts. So it's the provirus here, which is the name for when it's fully integrated, is almost 100% complete. It's missing two bases on either end. And you'll notice another feature here, these LTRs. These are long terminal repeats. And these are much longer than the inverted repeats that are on an IS element or a DNA-based transposon. These also have within them small inverted repeats, but the LTRs are involved in replication, not necessarily, they do much more than just provide a cut site for integration. You'll see how they are required in order to reconstruct the full length after it goes through this complicated series of events here. This part of the life cycle occurs in the cytoplasm, so you have an RNA that's in the cytoplasm. Either it's from an infectious particle. If it's a retrotransposon, though, it's just in the cytoplasm and becomes unpackaged. Reverse transcription, turning that into double-stranded linear DNA, happens in the cytoplasm, and then you get transferred to the nucleus and insertion. So let's look at, I've introduced very briefly here the idea of a retrotransposon versus a retrovirus. And you know the distinction is a retrotransposon, an LTR retrotransposon, does not make an infectious particle, but it does make a particle. That particle remains inside the infected cell, never infects an external cell. What we're going to do now is go through the life cycle of a full-fledged retrovirus. Everything is like a retrotransposon, except now its infectious particle is truly infectious. It escapes the cell and infects non-infected cells. So there are lots of aspects to this, and you see right off the bat, it seems much more complicated than a DNA-based transposon. It is. In the process, you'll see that these retroviruses use every trick in the book, everything you can think of, and how to jimmy the system, work it in your favor, misuse all the rules that are there, and exploit it for your own evil purposes. Retroviruses have figured all of that out and use it to the max. Notice how many different proteins they are able to make. 1, 2, 3, 4, 5, 6, 7, 8 proteins. 8 proteins. and they only have one transcript. Already, this is crazy talk, right? How can you encode 8 proteins in a eukaryote when there's no such thing as a polycistronic message? How in the world do you do it? Every way you can think of is how you do it. One way would be alternate splicing. Yep, it does that. Another way would be frame shifting. It has a stop codon, but sometimes the stop codon is not efficient. And it just jumps over the stop codon and continues in a different reading frame. What's another way? Oh, make a giant protein and then cut it up into small proteins, each with its own enzymatic activity. All of those strategies are employed in a retrovirus. Let's see how this, these different ways to cheat are exemplified here in the retrovirus. It's just not fair. Retroviruses just don't play fair. So the first thing to notice, the long terminal repeats here. We'll go look at these more in detail, but there's a region called R, and then on the right side of it there's U5. On the other side there's R, but on the left side there's U3. This is what you see integrated, and what you'll notice is it's missing, I mean, this is the form in the life cycle that from the RNA, the messenger RNA, you'll see the messenger RNA is not a complete genome. You don't realize that yet because the full long terminal repeat actually is U3, R, U5. U3, R, U5. And you see on this end the U3 is missing, and on the right end the U5 is missing. So in the messenger RNA, in order to make this messenger RNA, it has had to shorten itself, and in its process of replication, it is going to have a very devious and clever way to regenerate the two ends to make it U3 or U5. But before we get to that fascinating and headache-inducing mechanism, let us look at all the various ways that retroviruses rig the system. The first you see is from a single promoter right here. This is a messenger RNA. It ends up being spliced in two different ways. In one way, it's not spliced, the null hypothesis, and the other is this region here that encodes a central portion called Paul gets cut out. So you just have the GAG and you have the ENV. Well, the GAG, this is the three general clusters of activities. You have GAG, and you look down here on the chart, these are structural proteins, matrix, capsid, nuclear capsid. These are packaging proteins, not enzymes, but form a structural role to make the infectious particle, GAG. The central region is called PAL, abbreviation for polymerase. And you'll notice here, PAL actually encodes for much more than a polymerase. The only one that's really a polymerase is reverse transcriptase. It also encodes two other enzymatic activities. So PAL is more appropriately considered the region where the enzyme activity is encoded, whether it's a protease, an integrase, or reverse transcriptase. So you have structural proteins, GAG, the three enzymatic activities, PAL, and then ENV. To me, I don't know what that stands for. I think it must be environment. In the envelope, envelope, pecan, tomato, tomato. Envelope. I thought it was environment. I'm a biology major, botanist, combat botanist, yeah. Okay, envelope. And of course, wonder what that makes. Must make the envelope. The spikes and terminal proteins on the outside of the particle. Now, the actual skin of the particle is formed from the membrane itself. It buds off. All it's done has deposited these two proteins on the cell. You see a surface protein and a spike, a little cap to the spike. Okay, so frame shift, alternate splicing. So you'll get, in translation, you'll start off GAG and the reading frame will go right smoothly into envelope. And then that will get processed into two proteins. All the rest of it, it gets fused into one large protein, but what you'll see is it's making, in the processing steps, it is making a protease which knows to cleave the final products. So it makes a protease, and then the protease goes and cleaves the proteins that are being made so that they end up in these small units of activity. And it makes the reverse transcriptase and the integrase. So we've got protease having a big pre-processed protein, a pre-protein, and processing it down using a protease that's encoded by the virus itself. And the last thing is suppression or frame shifting at the end of GAG. In order to get a fusion of GAG into the Paul region, this is one protein, in this case you're going to have to get suppression of a stop codon, meaning the stop codon doesn't stop. It just continues to read through. Frame shifting is when it doesn't stop and switches reading frame. Both of those are used. So what do we have? Alternate splicing. We have frame shifting. We have suppression of a stop codon so it doesn't stop there. And we have processing of a large pre-protein down into individual smaller activities. Splicing, alternate splicing, the Paul is spliced out. Suppression and or frame shifting allows you to go from the GAG activities right into Paul. Processing takes a protease. And you see the resulting, the result is 8 different activities. Here you see a little detail on structure. Apparently this one, that says envelope, the 2 E and B regions here. But you can see the transmembrane protein is the stem, and the surface glycoprotein is a little ball on the top, and this determines the type of cell that it interacts with in the infection process. You see other structural proteins in here. You see a matrix protein that fills the center, and then you see a capsid that makes another a proteinaceous core, and then in the very core you'll notice that those little curly-Q red-orange molecules here, these are the infectious RNA. But there are 2 copies. 2 copies. That's very important because that allows recombination to happen. And you can have a helper virus rescue a defective virus. That's how you catch cancer. You have a viral oncogene that can replicate because it has a non-retrovirus, a non-oncogene carrying retrovirus packaged with it to provide the helper functions. the same strategy of a helper. You don't have to have all the enzymatic activities on the same molecules, even employed by retroviruses. And you can also see within the inner capsid, you have the protease, polymerase, RNase H, and integrase. Well, here's one we missed. RNase H is an additional Pol that the first figure did not include. So you've got protease, polymerase, reverse transcriptase, RNase H, and integrase as proteins are stored in here. So you already have these in place. For example, this protease that is needed to cut the first pre-protein into little pieces, you don't have to wait for it to be synthesized because it's already prepackaged. And of course, the membrane that you see here that these envelope proteins are embedded in, these are derived from the host cell in the budding off process. And here you see HIV budding off from the host membrane. Okay. So that's pretty complicated, right? But you just kind of stare at the slides long enough and you absorb it. You write it out, see whether you can recall the information there. The next series of slides is a bit complicated, and it's how you can go through these cycles of starting with RNA, then a duplex DNA, and then have that integrated, how you can regenerate, you can lose and then regenerate your long terminal repeats. These long terminal repeats can be 1,000 bases, and they're not inverted. They're not inverted. They are in direct repeat of each other. This differs from a DNA transposon in two ways. These are really, really large repeats that are made of sub-modules, U3 or U5. And they're not inverted. They are in direct orientation with respect to each other. On the very ends, there are still little inverted repeats, real small ones, and you still get, when it jumps into a new site, you get a duplication of the target, just like a regular transposon, 6 bases, 8 bases, 9 bases, as you repair the staggered ends. But the long terminal repeats are completely different than the inverted repeats on simple transposons or DNA-based transposons. So let's see, the RNA form of the virus, the linear DNA form of the virus, this is called the provirus, and the integrated form, well, which is called the provirus, the integrated form, isn't it? I don't have to wait till the labels appear. Reverse transcriptase, integrase, and this shows the long terminal repeats. In the RNA form, on the left side you're missing U3. On the right side you're missing U5. Notice you regenerate the full virus in the linear form. U3R, U5. U3R, U5. When it integrates, it's U3R, U5, U3R, U5. It's there, except you're missing 2 base pairs on each end. They are cut off by the integrase enzyme itself. It has a 2-base stagger when it cuts. You get 4 to 6 bases repeated in the target. And then within those, of course, where does it integrase? There are actually small inverted repeats on each end of the long terminal repeats. Although the LTRs are direct repeats. the linear DNA form contains terminal inverted repeats. Repeat, repeat. Please. I thought agrobacterium was bad. It's good, it's good. I wish we were back there in the old happy days. Okay, this is the slide from, what's the H word? Hawaii. This is the complicated part. So I'm going to try to make it understandable. The minus strand synthesis requires transfer to a new template. Okay, so minus plus, are we doing arithmetic here? The plus strand is the RNA that's packaged in infectious particle by definition. We're trying to make a double-stranded DNA intermediate. The first thing we do with that RNA is use it as template, that's plus strand RNA, template for making the first strand of DNA. Of course, that's minus strand. Minus strand happens to be DNA instead of RNA now. And then we'll have to go back, and that template RNA, which was plus strand, we'll have to get rid of that RNA and now make plus strand DNA, so that it truly is double-stranded DNA. Both strands are DNA, plus and minus. Okay. Nobody's left the room yet. Okay. You will in just a minute. Okay, so we got some little squiggly things going on here too. What is that? Somebody messing the slide up? Is that a hair on the lens? That is tRNA. It turns out that the host tRNA, one of them, is used to prime. Another devious trick is used to prime the synthesis. When you try to make that first DNA strand, which is the minus strand, it's primed by host tRNA. Now, you see this tRNA, it's standing on its head, right? The ends, the 5' and 3' ends, are up in the air. That's because the illustrator didn't understand how it works. He or she should have flipped this around and had it so that it's binding, and now you just elongate right here. You elongate this 3' end, which is sitting there, to make this second strand. Silly. Why didn't they do that? Because now that just confuses you. So let's see how this works. Oh, okay. A specific cellular tRNA acts as a primer for reverse transcriptase. Here you have the RNA that is unpackaged. This is plus strand. See, plus strand. Just drawn it on the bottom here. And you're going to start right there at the edge of U5. You're starting at the edge of that long terminal repeat. Notice this is the RNA stage. So the RNA stage doesn't have a complete long terminal repeat on both sides. It has a piece missing at the extremities. There's a binding site right here on the RNA that forms an RNA-RNA duplex with the tRNA. And then the reverse transcriptase, which is coded by the virus, but from the last generation, it was packaged like putting in an extra pair of socks in the suitcase. When it became unpackaged, there it was, ready to prime off of that tRNA to make this little piece of DNA, which is now minus strand DNA, and it goes all the way out to the end. This is called strong stop minus. And that's just experimentally, when researchers were studying the life cycle here, they saw they got this RNA that was always the same size. It had a very discrete start and stop. And it was to the left side, and they just called it strong stop because a lot of these things, they're going and you'll terminate at various places and you'll get sort of a smear on a gel. This gave you a really strong, distinct band, quite prominent, the first thing to happen. Strong stop, it's really making this left long terminal repeat as much as it can. Okay, so what's the next thing to happen? The 5' terminal region is degraded, it comes back, it chews off a little bit, that R portion is chewed back, and so you've got this. Now, what would you do next? If you, see, this thing doesn't have a brain. It thinks over hundreds of years in a chemical sense, as it responds to the environment presented to it at 1,000-year intervals. Another level of crystallization or consciousness. So anyway, what does it do? Well, it doesn't have good processivity on purpose. All the rules about what you're supposed to do, you know, learn to drive and get married and all that stuff, viruses just go, you guys do that. I'm just going to make as many copies as I can, and I don't play by rules. This little piece here comes apart and is floating around and hybridizes on the other side. Now, why can it? Because there's a direct repeat of the same sequences on the other end. So it comes off and binds to the other end, and now, see, it went all the way out, it did the R region. The R binds with R, and notice this R, though, has a U3 on the end of it. This is where it now acts as primer to go all the way across. Now notice what this has done. It has brought U5 over here on the right side. Where was this U5 originally located? It was internal. That internal U5 is now hanging off the end here. This is how it's regenerating its ends, regenerating these direct repeats by starting out on one side using the internal portion, and now it's hanging out like this. And now this can simply fill in, because this would be the 3', and it just fills in and repairs itself. So this now acts as primer, and it goes all the way to the end here. So we've figured out a way to reconstruct the right repeat, the right long terminal repeat, by taking this U5, because U5 is what's missing. See, it needs a U5, U3, but it doesn't have a U5. Takes that one, makes it, comes apart, slips here, and now uses that to prime. Pretty clever, huh? For not, it doesn't even know it's alive, and it's not even alive. It's just chemicals in a cell, in the dark. Doesn't even know what it just did. So we've pretty much got the minus strand. We're going to have to do a little trickery here on the left side to regenerate the left long terminal repeat, but we figured out using that scheme how to make the right long terminal repeat. Maybe we can just use the same scheme again to regenerate this end. If we could somehow get this U3 that's internal here, if we could just stick that on the end, by maybe making primers go back and forth somehow, we could do the same thing, couldn't we? And you're looking at it and going, yeah, it seems like it, but I can't quite make it happen in my head. And I have a brain. The next slide will show how retrovirus, which is not even alive, did this over, half a billion years. It figured it out. But it was the right answer. And this tRNA gets processed off. Let's see what I wrote up here. Note production of protruding 3' end. The viral RNase cleaves that back. That's that repeat region gets chewed back. That's also in the viral package. And then what you're going to get, single strand regions paired, then there's a jump, and then a reverse transcriptase reaction, synthesis of minus strand. Okay, so pretty much got we did a little repair over here. We've got the minor strand almost, and we need to get the other strand. How does tRNA act as a primer? Well, this is tRNA I got from someplace, and you see the 3' end on tRNA is the one that's protruding. A protruding 3' end? How strange is that? I mean, that's over and over and over again. Everything is the same. A 3' protruding end. If you just open this up, and you could use that maybe to prime reverse transcriptase. And so this is actually what happens. It opens up, forms a duplex here, and is used to prime minor strand synthesis. Okay. So you see here, this is where we left off. We have the first strand of DNA. Since it's complementary to plus RNA, it's the minor strand. Notice it's still needs some repair work here, but it's got now a U5 over here where the original RNA didn't have a U5. Here's the RNA down here below. Now you get a processing off of the tRNA that was used to prime. RNAs degrade, leaving fragments to prime DNA synthesis. So the tRNA is taken off, but now you start degrading the RNA from this DNA-RNA duplex. That sounds like an RNase H activity, doesn't it? And it just randomly comes in here and chews up most of it, but occasionally you'll get little stretches that are still left. Any of those little stretches can act as a primer. Usually it's the one that's closest to the right end, but it could be on here, you could extend it all the way to the end. There's no one particular place that it primes. It's just a random priming by these little degradation products of the RNA that are left in place on that newly synthesized partial minor strand. Well, let's take this one. It was used to prime, and now it goes all the way out to the end, including the U5 element that's just hanging out there in space. You've totally now regenerated the right end. And you have, it's primed here from random RNA that's left over from that infectious plus strand. But now how is it going to get all the rest of this stuff? Now it's probably become obvious to many of you. The same little trick. This is going to fall off and join right here. It has taken this internal U3 and moved it so it's the external portion of the LTR to regenerate the left LTR. And of course, that is used to prime on out again. And then this is a simple repair to come out here. So you see the same general strategy where you form a little bit of the left or right side and it floats off and is used now to do two things, to capture that internal portion of the long terminal repeat and stick it on the end, and to act as a primer itself to complete the synthesis of the respective strand. See if you could sit down with a blank sheet of paper and think like a virus. You don't have to cut the lights off and draw it in the dark, but see if you could duplicate both your strands and regenerate those beautiful long terminal repeats that you're so proud of, like a cat with its fluffy tail. Restore them. Use trickery. Okay, Strand transfer causes recombination. Reverse transcriptase, of course, was involved in both of those strand jumping reactions. That property of reverse transcriptase, to not stick too tightly to its template and to move with its newly transcribed product to some other place and prime again, also leads to recombination sometimes. Because if it jumps to not a copy of itself, but jumps to a nearby copy, except this is a copy that may be defective or be a little different. It doesn't necessarily jump back on its same molecule to prime. It comes off and it can jump wherever there's a template available. So this way you can get fragments from one virus being transferred to a second virus by that strand transfer property of reverse transcriptase. You see it dissociating, and here it's going not onto the other end of itself, it's just jumping to a new template. This mechanism is called copy choice. So this is a way that fragments of the viral genome are combined that does not involve homologous recombination by the host. This is all generated as a property of its own mechanism of copy choice. It jumps off and goes to its own template or to a nearby template, a new template. Copy choice. Okay, that's as complicated as it gets. That may be as complicated as the entire course gets. That's it. And you understand it. You won't understand it when you walk out the door, but right now you understand it. It's fading fast, but you think you understand it until you get that blank sheet of paper and you go, I don't understand it. But right now, in this bliss of newfound knowledge, it's all still happy time, right? We know this. This won't take much to brush, and it won't. Just go back and look at it and be able to replicate your own viruses in case they get in trouble. Okay, so this is a diagram you don't have to memorize. Just know generally what's happening. And this is how the terminal, long terminal repeats interact with the integrase enzyme. It forms this structure that you've seen before in other transposons. You don't really need to memorize this. Just know that there are two bases missing. See the two bases here? Two bases missing at the ends of the long terminal repeat when it integrates. Leaves a 3' recessed end. Okay, so that's how replication occurs. And you see how it needs an RNA and a DNA phase to make it work. The RNA part is infectious, but its long terminal repeats are not complete. But the missing part is kept in the other one, in the internal region. This one is kept in the internal region. And then when it makes itself linear, double-stranded linear, the only truly intact form of the virus, before it jumps in, it's intact. And then when it jumps in, it loses 2 bases on either side again. So that's replication. How is it expressed? What kind of transcription control does it have? Does it use its own RNA polymerase? It certainly has its own replication enzyme, a reverse transcriptase. But it turns out it uses the host RNA polymerase. And there's several forms of these, but sometimes it uses Pol3, sometimes it uses Pol2. We haven't talked about that yet, and some of you, I'm sure, know. In eukaryotes, there are a minimum of 3 regular RNA polymerases, and they are devoted to their own subset of genes. You have one that's devoted to making the ribosomal structural RNAs. That is RNA Pol 1. Then you have RNA polymerase 2 in eukaryotes that is devoted to genes that encode proteins. And then you have RNA polymerase 3, which is devoted to those genes which encode tRNAs and other small nuclear RNAs. So the host enzymes, whether it's Pol 2 or 3, is used to make the copy of RNA. So this is the integrated virus, integrated form, missing 2 on either side, but you remember when you make the RNA, it's going to be missing this whole U3 over here, and it's going to be missing this entire U5 on the right side. So the fact that 2 bases are missing on the extremes is irrelevant. It could have lost the whole thing. Where does it start transcription? It has a eukaryotic promoter in U3. Notice that U3's here internally. Well, here it's externally, but the promoter part, this is this is the internal U5, and here it's internal U3. But the U3 that's at the extreme left terminus, these 2 bases didn't matter. It still has a promoter that controls RNA transcription, the start site for transcription to make that plus strand RNA. Now, it could start here, or it could start here. If it starts here, it just makes this repeat and goes into the host genome. That would be sort of a dud. That would do the virus no good, but sometimes it happens. And that's how these retroviruses sometimes pick up host genes. In fact, host oncogenes, depending on where they land. They pick up things that can cause cancer when they start jumping again. But this is the normal start. We don't quite know why it uses this one and not that one, because it's exactly the same sequence. U3 is U3. It's a repeat after all. But there's something there, binding proteins, we don't quite know, but it always, for the most part, not always, but the vast majority of the starts of transcription are at this U3, not that one, except when it is that one, of course. So let's see what happens. So you get this long transcript, and then you get the different kinds of processing, and you see the transcript itself has U3 internal, U5 internal, but each end is missing. Reverse transcriptase, and you generate the full long terminal repeat. Sometimes this results in activation of host genes that cause the tumorigenic state when it starts over here. But in terms of its own replication, it doesn't need to start over here at all, it just needs to start at the left U3. Retrovirus may transduce, which means trans again, to carry, to move. It may transduce cellular sequences. How much more time? I'm out of time. We will finish this up, and I will go back and hit the main points here of what you need to know in terms of the exam, how to keep this straight in your head next time, and then we'll go back over everything, and if you got questions, you can ask me in class next Thursday, and if you don't, I will sort of revisit some key points also and help you on the exam. See you Thursday.